old 7-year cycle thing, meaning that all the topics come up in 7-year cycle. So I actually went back into my notebook and looked in what did I talk about 7 years ago, and it was exactly that kind of topic. So most of what I'm presenting today is actually old stuff, but it might be useful, and looking into the many new and young faces in the audience, it might actually be useful to talk about this. So this old stuff actually led in the last years, and it had been mentioned several times also during these presentations, this session, to some of the most cited phenotyping publications ever. So think about your old stuff before you go to the only to the new one. The second thing which I wanted to also convey in this session is that controlled environment is not only helpful for breeding, there is also another dimension which is about getting mechanisms more detailed, and that's also something where controlled environment can help a lot. So this is the two different kind of things which I wanted to contribute to this session. So the first one is which kind of environmental conditions should we actually choose, should we actually measure. And I think Thomas at the beginning in his talk already mentioned some of this work which we did here on metaphenomics. So this is a meta-analysis on many different publications, and essentially what the meaning of that whole thing is that we compared the growing conditions of Arabidopsis, of herbs, of woody species, and how they are actually grown with respect to photon availability, so light conditions, and temperature, and how they are actually done in controlled environments. And essentially what this delivers is this kind of setup here. So you can see a mean temperature where if you go through all these publications where they're grown, you can also see a mean daily photo integral of how much light they actually were received during that growth conditions. And then you can compare these growth conditions in the growth chambers or in the greenhouses with what actually they experience in the field. And this is what we did here. And there are two main messages, a complicated picture. I don't want to go into all the details. Essentially, it's comparing in these squares on the right, it's comparing the different conditions in which plants are grown, gray, yellow, and green. You can read this down here. And on the other hand, it's looking into in what kind of environment with respect to temperature and with respect to light conditions plants are actually growing in the real environment out in the field, in the tropics, in Arctic conditions, in temperate conditions. And the first thing which you see is that plants are exposed to a wide range of different temperatures, so it's not constant temperatures over the entire vegetation period, not talking about day-to-day variability even in here, and that these sections which we have here, this kind of quarters, which squares and rectangles which we have on the right, they are always on one side of the vegetation period, and they're not fully covering the entire vegetation period. So the question is, do we actually grow the plants in the conditions which they're actually out there, or do we expose the genetic system, which a plant is, in an environment where it somehow functions, but it doesn't function the same way as it actually does in the field? And essentially the outcome of all this kind of analysis is in here. Essentially we usually grow the plants at too high temperatures and too less light. Very simple. The plants still respond. They are responding nicely to temperature and light, and this is the background slides which you see in the background here. And the way how we are treating them, they're actually in a part of the response curve between temperature and light where we really get significant changes. So it's not something which is irrelevant, but it's really a relevant part of the response curve where we expose the plants to in a part which is not really simulating what we actually see in the field. So that's one of the simple messages, that we usually in the growth chambers expose plants to less light and to too high temperatures compared to what we actually get in the field. The second part, which I want to focus on, and I think Thomas also mentioned before that one should link up with ecophysiologists. So in my background training, I was an ecophysiologist, so I think that's a thing which I can contribute here. And one of the important things where ecophysiologists are faced to is that plants are dynamic beasts and that also environments are dynamic beasts. So how are dynamic and heterogeneous systems actually working, and what are plants actually exposed to? Plants are never exposed to homogeneous light conditions. So even if what we saw today already from heterogeneous light conditions in the greenhouse, this is usually talking about light conditions from above ground at a canopy level, but what happens inside the canopy is completely different. This is completely different with respect to the heterogeneity in space and time. So we have fluctuating light conditions there, so the plants really have to respond to fluctuating light conditions in an optimal way and not only to constant light conditions. So that's a challenge for the plant. So think from a physiological point of view, the plant needs to optimally extract energy from a system which is very fluctuating in space and time. How do you do this best? That's the physiological question behind there. And the same essentially is below ground. I took that picture, which it's really beautiful, thanks Michelle, to see how intimate the interaction between the root system and the soil is. And this is not just something like which is looking like agar, it's a really complex environment around there. So also below ground, we have very, very different environmental conditions in space and time. And the question now for this kind of controlled conditions is how complex do we have to generate these environments in order to really see a performance which the plants actually have in the field. And if you look in this kind of table, which is just a simple table which I made also 7 years ago, showing how different the life environment for roots and shoots is. It's essentially extremely different for essentially all the parameters you can think of. So do we have to, for example, change all these physical characteristics? Do we have all the heterogeneity of physical characteristics, the chemical characteristics? Do we have to all provide them to the plant in order to get a useful result from our control environments which we can transfer to the field? That's a completely open question. It's not solved. But we know that there is this kind of complexity. We know that there's a lot of complexity with respect to light conditions. We already saw that before. Light conditions fluctuating because of external light in, but that's the real life of a plant. It's not something which we generate as a kind of complexity. It's out there. And again, this is just the light regime above the canopy. In the canopy, we are talking about very different conditions. And at the same time, we have the plant itself being a dynamic system, and this is also, sorry for the old guys who know that already, very old stuff which we did some years ago, looking into dynamic of the plant itself, so dynamics of plant growth. And I don't want to go into details here. Essentially what we can see is how dynamically growth is actually inside a leaf, and this is with over a few hours. And the exposure of that leaf to light is heterogeneous. The plant itself has different spaces on that leaf which are exposed to different external environment. So how much kind of complexity do we have to achieve in order to get some useful results? And there are some strategies in there. So the plant, to some extent, in above ground for dicots, has a strategy to actually uncouple itself from these environmental fluctuations by running an internal program. So this is a complex picture, which I also don't want to go into detail, where we can see there is essentially a kind of endogenous clock rhythm of growth rates in dicot plants, which is a way to uncouple itself from heterogeneity of light conditions from the outside, or fluctuating conditions from the outside. We can even go extreme, and that's, I think, still a very nice one. You can actually punch out leaf discs, let them grow on fluids and provide them with nutrients, and they grow exactly with the same kind of diurnal curve. And you actually, after some days, you can fit them into the hole, and it still fits. So this is a very simple indication that the program is running there, and that we have something which is where the plant is somehow trying to uncouple itself from the environment. Very nice for dicots, different in monocots. So leaf growth in dicots is very much controlled by these endogenous rhythms, and there is an application for the environment is done, the adaptation of program of the leaf growth itself. And this also gives opportunities to go into deeper understanding with mechanisms, because if we have these kind of maps of physiological processes on the leaf, we can actually take samples from there, so what we call guided sampling. But we can also go below ground, and I mentioned I had this picture before. It's very diverse with respect to from above ground, with very different from above ground with respect to spatial patterns, temporal patterns, chemical structural environment, mechanical properties, very, very different below ground. And it's hard to imagine how to get useful information from these kind of other things in pots or in agar plates to these real conditions are there. But you can study mechanisms. You can study what kind of exposure do we have. Temperature, for example, temperature in the soil is varying with depth. They are varying with daytime. So do we have to simulate all of this in the greenhouse in order to get that kind of information, to get a better performance in the field? Do we have to look into the heterogeneity of the soil? In the field, we have heterogeneous soil with respect to, in this case, for example, nutrient concentrations on a mapping of nutrient concentrations. This is changing over time. So this is the same patch 3 days later. So we have a huge heterogeneity and dynamic in the system there. How much of this do we have to actually simulate in the greenhouses to get something useful for the field? At the same time, we also are aware that the roots are actually changing their environment. So this is what you see over there, for example, is the pH or oxygen concentration, and you can see at the end of this one that this is actually on the right, this is actually where a root is growing through. So it's not that plants are just accepting the environment, but they're also changing the environment. So how much of this do we actually have to monitor on a smaller scale? Also on the root side, we can use this control environments to get information about mechanisms. In this specific case, we grow them at stable temperature conditions, and what you see is that you don't see a diurnal course. So it's very different from above ground, and this has good reasons because the root is very strongly responding to the immediate environment. So within seconds, it can actually change its growth rate with respect to temperature. So this is a completely different system compared to above ground. Above ground, we are buffering, we are uncoupling from variation, while below ground we're having a very strong response, and that makes sense from the physiological performance of the root system. We can go into more details there. You can see this kind of temperature changes in different species, very, very high accuracy there. And we can also see that temperature gradients do make a difference, and that's an old experiment, also an old experiment which we published some years ago, where we simulated temperature changes, temperature gradients in the soil, actually between 10 degrees at the lower bottom and 20 degrees at the upper part. So 15 degrees is the kind of median median temperature. So these three things here, this is the root and shoot biomass at constant temperature of 10 degrees, constant temperature of 15 degrees, and constant temperature of 20 degrees. And if you expose the plant, that's barley, into a temperature gradient between 20 degrees at the top and 10 degrees at the bottom, you get that. So this is the same barley plant 6 weeks later, 6 weeks old, has a completely different biomass. So they're immediately responding to the temperature gradient. Do we have to build temperature gradients in all these controlled environments? I don't know. It's an open question. So this is a kind of information first on the below ground, above ground. And this is the last one, which I just wanted to show, a last one which shows that we have a strong interaction between above ground and below ground. That's also often neglected. This is an adaptation of the root growth to changes in light environment above ground. And this is at stable temperature above ground, below ground, but just changing the light conditions, and you can immediately see over hours and over days how the adaptation of the root system actually works. So it's a very dynamic system, and the question would be how much of these dynamic systems do we actually have to simulate in controlled environments in order to get something out decent for the field. So take-home messages, simple ones. Controlled environments can be used to run plant systems at relevant environmental conditions, whatever this means. Not clear so far. If you are interested in comparison of field conditions to in-house conditions, but they can also be used to expose the plant to artificial conditions in order to dissect mechanisms. That's a thing which we usually don't talk too much anymore in phenotyping background, but it's a valid question. And the second part, heterogeneity and dynamics of root, shoot, and environments are very important for plant performance. They are extremely different with respect to dynamics and spatial and temporal heterogeneity. And the heterogeneity is a biological issue, because essentially that's the reality which the plant has to cope with and where they actually have adopted to during evolution for millions of years. And on the other hand, it's an experimental design topic. How much of this kind of dynamics do you have to build in your experiments in order to get something decent out of it? Thanks very much.